crystallized tree sap, which we call amber, waited for millions of years with the mosquito inside until Jurassic Park scientists came along. Using sophisticated techniques, they extract the preserved blood from the mosquito and, bingo, dino DNA.
No, that's not how it's done. Because any piece of amber, no matter how pure and translucent it looks, contains all kinds of microscopic bits and pieces of the past. Pollen, midges, nematode worms, fragments of twigs and petals. All of these things have their own DNA, and if you drill a hole through the amber and insert a needle, you're going to pick up DNA from any of these other life forms that are not dinosaurs.
Cut around the circumference of amber using a small circular saw. Then wash the nugget liberally with alcohol to get rid of dust, grime, and DNA-containing material. Move to a clean laboratory and with a clear plexiglass glove box, put all the equipment you're working with and switch on the UV light to remove any DNA on the equipment. Then turn off the UV light and put the amber piece into the box. Remove the materials in the box into a different room.
Plunge the amber into cold liquid nitrogen to make it more brittle still. Carefully crack open the amber to expose it.
Label test tubes to organize the parts of a mosquito. After test tubes are sealed, open the plastic block and put instrument in the seal to be cleaned. Again, you move to another lab contaminant-free. You then break the cells open to get the DNA by using soap detergent to disrupt the lipids binding the cell membrane. Add an enzyme that degrades the protein in the resulting solution.
You then add phenol to the mixture. To extract as much DNA as possible, shake the test tube to mix the contents. Then spin the test tube in a centrifuge to separate the water and phenol. Suck water layer with a pipette and remove as much as you can without sucking any other layer. A couple of teaspoons of cesium chloride crystals and dissolve them in the small amount of water as possible to make a slop of cesium chloride. Spin the solution in a device called an ultracentrifuge. Before adding the insect DNA to the test tube with a salt slurry, add a dye that's designed to chemically attach to the molecules of DNA. Shine a UV ray on the test tube to get a red glow from the dye. Finally, extract the DNA by sticking a fine needle through the side of the test tube.
Clean laboratory and with a clear plexiglass glove box, put all the equipment you're working with and switch on the UV light to remove any DNA on the equipment. Then turn on the UV... Cut around the circumference of amber using a small circular saw.
Take apart the creature with a microscope and pluck out the muscle tissues and quickly drop it into the test.
In the solution in a device called an ultracentrifuge.